Now, I'm going to segue now into the actual research component of this presentation. So the focus for my talk today is, broadly speaking, really to describe how we've approached our goal of understanding the causes of impairments in autism spectrum disorder. And for today, I'm going to use autism spectrum disorder and autism interchangeably. But understanding causes of autism spectrum disorder oftentimes is overwhelming itself, is a tremendous challenge. And it does feel like we're looking for these small little needles in haystacks, or to use another analogy, we're trying to go backwards from a tornado occurring in Iowa to the initial butterfly flap in India. For those of you who don't know what I'm talking about there, the butterfly effect was originally described to suggest that tiny changes in wind patterns in one part of the world can lead to dramatic shifts or events elsewhere. In other words, seemingly trivial events ultimately can result in something with much larger consequences. They have a nonlinear impact on very complex systems. And as we think about autism, we're trying to go back from a complicated clinical picture to what often are referred to in genetic terms as non-significant events. And building that bridge and understanding that process, and then being able to use that knowledge to actually understand or better predict where areas of support are going to be needed for individual children has been tremendously challenging for the field. Before I launch into some of the work that has been done in this realm, I want to first address what is a fair question. Why do we care? Why do we need to know about the causes of autism? And this is a question that I think has been fairly posed by self-advocates, by other researchers, and frankly isn't quite as easy to understand at times, or easy to describe at times as maybe we initially hoped. But the reality, my perspective here, is that we're trying to increase knowledge of individual development. We're trying to understand mechanisms that contribute to individual developmental patterns, identify or understand them early in ontogeny, early in child maturation, with the goal of providing more effective supports. We're also trying to advance more individualized strategies to mitigate some of those symptoms that may cause suffering in individuals with autism. And by understanding the neurobiological or etiological processes that contribute to these challenges as children develop, we may be better able to support those children and help them through development in ways that are much more conducive to better outcomes. All right. So if we start digging into this question, though, of how we get from these small events to this tornado, if we approach this from a behavioral or a clinical perspective, we can describe autism or start to think about autism not just as a spectrum, right? I mean, we often hear about autism is a spectrum, and from severe to mild, but in reality, it's far more complicated than that, right? Everybody who's worked with individuals, autistic individuals, knows this. It is a spectrum of different traits, right? There are many different abilities that are variably impacted in autistic individuals. They're variably affected across individuals and then within individuals. So in other words, and I think I'm preaching to the choir here, but an individual with autism that's identified as severely impacted may have relatively unaffected or intact abilities and development in some of these traits listed here, okay? So it can vary within individuals, it can vary across individuals. And so if we start to think about this as just one entity and then look for the causes of this one entity, we're really glossing over a lot of details and embarking on a search that really isn't directed appropriately. This same complexity is echoed when we look at neurodevelopmental processes that are associated with autism. So this was a really cool study that was just looking at 16 different mouse models of gene variants that are highly penetrant and associated with autism. So each of these genes listed here within these clusters has been implicated in autism, but actually implicated in population of individuals and shown to be highly penetrant. That is, if you have this variant, you have a significantly elevated risk of autism and IDD as well. And what they did is they looked at these different mouse models and interestingly found that relative to wild type models, there were clusters that each showed very different patterns of atypical functional connectivity or communication between different parts of the brain. So importantly here, not only did these different clusters show deviations in different brain systems, but they showed different patterns of deviation. Some were hyperconnected as represented by these warm colors, some were underconnected or hypoconnected as represented by the cold colors. So if you think about that for a second, if we think about autism as just a single entity, which often we talk about it that way, but I just explained how it's not. But even if we just allow that for one moment, what this suggests is a great deal of equifinality in psychological terms, right? That is that we're getting to the same clinical definition or clinical label through very different biological pathways, through very different patterns of brain development. If we go a level further, Merlin Butler and Ann Manzardo published this paper years ago, and they identified close to 800 different genes across all of our different chromosomes that have been associated with or implicated in autism in some way. So there's a lot. The most common known cause of autism is genetic, and more specifically, inherited gene variants, both common and rare. Common making up the majority of those inherited variants that contribute risk to autism. All right. So autism is complex. It's etiologically complex. There are environmental factors that contribute to risk that may interact with these different genes that also contribute to risk. And then within the genetic background or the genetic landscape, we have inherited common and rare variants, as well as these de novo variants, right? These de novo variants that weren't present within the family lineage. But it's highly heritable, and the majority of cases are believed to be attributable to inherited gene variants. Importantly, those cases also are attributable to common gene variants. And what does that mean? These are common variants that have very small effects. So these incrementally additive allelic variations each contribute relatively low risk to autism, right? So this is just above 1. An odds ratio of 1 means there's no risk whether you have it or not, right? So these odds ratios are just above 1 for each of these different common gene variants identified or associated with autism. But the key here, though, is that if you have multiple of these variants or the right combination of these variants, eventually it can push an individual over this threshold and into this categorical pattern consistent with an autism spectrum disorder diagnosis. And so this idea that multiple common variants combine, interact with, or add upon one another to lead to autism is the idea of polygenicity. Okay, so there's polygenic, many genes interacting, working together to contribute to or underpin an individual case of autism. All right. For those of you that are thinking statistically about this, you might be looking at this and say, well, the way to approach it, the only way to approach it, and this has been a common way, is we're going to need thousands of individuals, right? You have a really small effect. So how do you detect that small effect? Well, you just add more people to the sample. And that's true. And that's what these GWAS studies have done. These genome-wide association studies have done. And we get in 10, 15, 20,000 people, and geneticists are really good at sharing data, right? And so that has yielded some of this knowledge. And that is an important approach in this context. But for those of us that are trying to approach this in different ways, or maybe trying to advance more quantitative and intensive phenotyping strategies, characterizing 15,000, 20,000 people is a major, major difficulty, right? So how else can we approach it? Well, one way that we can think about this is by trying to understand or identify those traits or those factors that lie intermediate the complex behavior or combinations of behavior that are autism and the genes of small effect that contribute to risk or lead to autism, right? And so some people have referred to these as intermediate phenotypes because they are intermediate the behavior and the genes. I'm going to talk about them as endophenotypes. As a specific type of intermediate phenotype, is a specific type of biomarker that is specifically linked to genetic underpinnings. And these can be at multiple different levels. These are just arbitrary units of analysis that I thought to put in this figure, but they could include measurement at the neuropsychological level, at the level of brain structure, at the level of brain function, circuits, cells, synapses, all of that, right? So there are multiple levels here, but the idea is that we're trying to really increase statistical power for identifying associations with these small effect genes by studying autism at more intermediate levels more proximal to the genetic origins. So this is a concept that Irv Gottsman brought to the field of psychiatry 50 years ago. He took it from plant biology, okay? And has utilized this, and other fields of psychiatry and other fields of medicine have really leveraged this concept and this approach to better understand the neurobiology of disease, or the biology of disease, I should say. Endophenotypes are quantitative traits associated with genetic liability. And their power is that they allow us greater ability to detect genes of relatively small effect than if we just look at affectation status or even behavioral manifestations that are much further downstream alone. And when Irv Gottsman described endophenotypes, he suggested that they should be quantitative. They need to be dimensional. We're not talking about categorical entities. You either have it or you don't, right? These are quantitative dimensions, quantitative traits. They're associated with the illness and outcome. Goes almost without saying, but they should be associated. The more severe you are on these particular traits, the more severe you should be on the core features of a particular disease or disorder, or disability, or outcome. They should be independent of clinical state, right? We're looking for endophenotypes that are not dependent on how much sleep you had the night before, right? Not dependent on how interested you are in doing that particular test. But these need to be really objective, reliably measured traits that are identifiable, detectable across different states. They should be heritable. That's what we're talking about. We're talking about inherited factors that contribute to or cause autism. And as a result, they should be evident in unaffected biological relatives. And in fact, they should be evident in biological relatives to the degree to which that individual shares DNA with the individual case, okay? So second degree relatives are less likely, should show less severity of these endophenotypes than first degree relatives, okay? First degree relatives should be less affected than individuals that are cases or probands. All right, so it's a good idea. It's really worked. It's really been useful in psychiatry. It's been useful in understanding psychosis, so adult psychiatry. It's been useful in understanding cancers, heart disease. Developmental disabilities, we haven't made quite as much progress in this regard. So there aren't really any established endophenotypes within autism, but there are strong candidates. Hyperserotonemia is one that has been studied for the past 30 years as a potential endophenotype and seems to be present in a subset of families affected by autism. John Constantino wrote a nice review of this in 2018 and really proposed 4 strong candidates, and this is not an exhaustive list to be clear, candidate endophenotypes that each contribute independent risk, serve as independent predictors of autism. Okay, and that's an important concept. I'll come back to in just a moment. And each of these is reflective of some polygenic mechanisms. Okay, and so you refer to them as developmental substructures of autism. And so the idea here is that these endophenotypes, you only need maybe one of them, right, to contribute to be at higher risk for autism. They're each quantitative traits, social visual disengagements, how much individuals are looking at the faces based on Ami Klin's work. ASD specific background, that is to say that family members show broader autism phenotypic traits. They don't have autism, but they show characteristics that parallel the core features of social communication, impairment, repetitive patterns of behavior as autism. They have autistic traits, but they don't have autism. And families with these backgrounds or broader autism phenotypic backgrounds tend to have slightly elevated risk for a child with autism relative to the neurotypical population. But then he identified motor coordination and ADHD trait impulsivity. So these are each features that are sometimes referred to as comorbid, sometimes they co-occur in autism, but they're not core features of autism. This is the developmental substructure piece. The idea here is that you have these endophenotypes that reflect some sort of pattern of atypical neurodevelopment that leads to or reflects the mechanisms that lead to autism. And in this model, if you think about it, what he's really saying is that developmentally, autism doesn't look like autism in some people. Autism in young children, in some young children, may be motor coordination impairments, may be ADHD trait impulsivity that turns into autism overdevelopment. Okay. Importantly here too, one last point on this slide. It's a beautiful slide, but I have to move on at some point. The other main point here and key concept is when we study people with autism, we're studying a biased sample, right? All these people have autism, all these cases have autism. But some of these cases, some of these individuals don't show these patterns of social visual disengagement. Some of them don't show they don't have family members that show these broader autism phenotypic characteristics. So they do, but they still get autism. They still get it from somewhere. Now, it could be from rare variants, it could be from some de novo variant, it could be some environmental contributors. But the point is that these individuals may just get it from these non-autism-like endophenotypes, neurodevelopmental mechanisms, all right? And I bring that up because prelude to later, our data is suggestive of this, all right? Okay. So we've been thinking about these ideas for a while. This is back, some of this data is back from my postdoctoral training, when I thought I'd be done with this whole question by now, but I'm not. But the idea is that we started studying different sensory motor and cognitive control systems, primarily because, well, personally, I had a strong appeal to things that were quantitative, that I could feel confident in, right? These are highly quantitative measurements, they're highly replicable. So these happen to be core components of a strong endophenotype. And we started measuring these systems using different oculomotor tests. All right, so I think many of you have seen these measured before, but indulge me for just a moment. But we're presenting individuals with stimuli, including central fixations, and then peripheral targets just show up. We don't have to provide them with any instruction, right? Our eyes naturally, if we're in a dark room with no light and all of a sudden something flashes in our periphery, we make an eye movement towards it. In fact, it's very hard not to make an eye movement towards it. So we capitalize on that. And we can also look at how well an individual is at inhibiting a movement or a behavior or a response that we don't want them to engage in. So we give them the same stimuli, but we provide the instruction that when that peripheral target shows up, don't look at it, look in the opposite direction, and we refer to this as an anti-saccade. Okay, the saccade is just a rapid a shift in eye gaze, and anti-saccade is a shift in eye gaze away from a target. We've done these studies, and these findings have been largely replicated both by our own group and independent cohorts, as well as many other groups who have been studying these different systems. And what we see is that individuals in autism tend to show less accurate or precise saccades. Here you see the saccade error in degrees of visual angle, and that is greater in individuals with autism. And they also make these anti-saccade errors in which they look at the target instead of looking away from the target. They don't inhibit as well. And they make those anti-saccade errors at a higher rate than their non-autistic neurotypical peers. Okay, so it's been demonstrated and replicated. And I should say also, the rate at which individuals make these anti-saccade errors appears to be associated with the severity of their restricted or repetitive behavioral impairments. In other words, the extent to which an individual can inhibit an unwanted behavior is related to the rate or the severity with which they exhibit contextually inappropriate or repetitive patterns of behavior. Okay, so we think about this as sort of a quantitative neurocognitive mechanism associated with this clinical trait. But it's not an endophenotype, right? An endophenotype is something that gets us closer to understanding genetic underpinnings. So a while ago now, we looked at these same behaviors or abilities in unaffected parents and siblings of autistic children. And we found multiple different oculomotor differences amongst the parent and sibling samples. But to be concise here, the same patterns that I just showed you in the probands or autistic individuals, including increased error or reduced accuracy of saccades, as well as increased rates of inhibitory control errors or increased anti-saccade error rates, each also were demonstrated by the parents and the siblings. Okay, the effect sizes were smaller than we saw for the probands, as we expected, but it suggests that there's some sort of familial transmission of these different oculomotor abilities that are affected in autism. So since that point in time, we've extended these studies. We started to look at other behaviors, other motor systems, quantitative motor behaviors. Most of you have probably seen me present on this or seen someone from our lab present on this particular paradigm. So I'll go through it relatively quickly, but in short, individuals are pressing on two buttons or transducers with their index finger and thumb. When they are pressing, they're viewing two horizontal lines, a red line and a white line. The red line will turn green, telling them to go and to start pressing. And when they do that, the white line will move, and it'll move higher with increased force. And their task or instruction is just to maintain that force line as steadily as possible at the level of the green line. Over here, you see sample force traces. This person is quite steady. This person is more variable. Their force level is going up and down. It's not moving as much because we've changed the visual feedback parameters. That's for another talk, okay? But the key here is that what this is really looking is how well an individual can utilize visual feedback information to maintain a precise force, output, or motor behavior. This is the first study that we published on this particular experiment or task. As you can see from the line below, we've now replicated these results multiple times. There are several parameters that seem to be different in autism relative to neurotypical peers. The key that I'm going to focus on is that individuals with autism show increased variability during this task. In other words, that white line is moving up and down much more than individuals without autism, okay? We see this vary in terms of its severity, depending on how hard they're having to push. This is a percentage of their maximum voluntary contraction or maximum force, depending on how much visual feedback we're giving them. But the key here is that variability is increased across these different task conditions. One of the studies that we published on this, separate from what I just showed you, so a separate sample, We did EMG recordings while individuals were engaging in a very similar task, except just pressing with their index finger, recording from the first dorsal interosseous muscle right here and looking at motor unit firing. A lot of nerdy terms in there, I apologize. But the key here is that when we look at the neurophysiological underpinnings of force control during this type of task, we see that the mean discharge rate is overactive or is elevated in autism. And then importantly here, that biology, that physiological difference, which is a highly reliable measurement, is also highly associated with the severity of autism. So now we're talking about the biology of a behavior that is not an autistic trait or a core autistic trait, but the biology is predictive of the clinical severity. We also see this at the central level. So we can do the same experiment with our colleagues over at Hoagland Biomedical Imaging Center. Bill Brooks was here. He's gone now, but Bill's been a great collaborator, as well as Laura Martin and Rebecca Lepping on these studies. And we've studied these visual motor systems that have been pretty well mapped via primate studies, as well as human lesion studies. In short, when we're doing this kind of activity, when we're getting visual feedback, we're processing visual information V1 or primary visual cortex, and that information is relayed along two different pathways: the dorsal pathway to the superior parietal lobule and parietal cortex, posterior parietal cortex, and the inferior parietal lobule in the temporal, IPL, as well as the temporal cortex. And temporal cortex, this ventral system is really allowing us to differentiate object features. It's called the what pathway. The dorsal pathway is referred to as the where pathway, and involves both superior and inferior parietal lobule. These systems then translate information to M1 or primary motor cortex, as well as dorsal premotor or ventral premotor cortex through this cortical route. There also exists a subcortical route in which spatial information from parietal cortex is relayed to cerebellum. Cerebellum provides corrective adjustments in response to the error to adjust the motor output via primary motor cortex. In other words, cerebellum is taking in that error information, the space between the horizontal lines in the experiment I just showed you, and it's trying to reduce that spatial error by adjusting how much force is being generated through the periphery and as commanded and controlled by primary motor cortex. So we utilize a fiber optic system to conduct experiments very similar to what I just showed you in our laboratory studies during functional magnetic resonance imaging. We believe, and there's strong evidence here, that task-based fMRI studies really give us greater power, much greater power to understand physiology related to specific traits, as opposed to looking at just task-free or resting state studies. So it's been important for us to use these task-based paradigms, and I think they've been pretty powerful. This is data that Catherine Unruh has been analyzing and leading and working on using a paradigm similar to what I showed you in the magnet. And what's critical here is really the connectivity differences that we see between cerebellum and posterior parietal cortex on the top figure. All right, and so I just showed you that visual motor network. That posterior parietal cortex is providing spatial error information to cerebellum, which is then being used to adjust the motor output via the motor system and primary motor cortex. And this connectivity between posterior parietal cortex and cerebellum is greatly reduced in autism. And you can see that here. In fact, overlap between the two different groups is relatively minimal. Okay, so again, now we have a biology of what we're seeing as a potential biomarker, but we still need to understand whether it's associated with the disease itself. It is because we see it in autism, but also because it seems to be strongly associated with core features. So the ADOS CSS, many of you are familiar with, but it's the ADOS, Autism Diagnostic Observation Schedule, one of our core diagnostic assessments for autism, calibrated severity scale. So this is intended to be a dimensional measure of severity of core autism traits, and the less connectivity between posterior parietal cortex and cerebellum within an individual, the more severe core autism traits appear to be amongst this particular sample. This isn't really halftime. This is like 3/4, I promise. But to summarize where we are with this information, right? So we're trying to establish endophenotypes. It's cool that we can find these quantitative tests that are different in autism and controls. Are they quantitative? Yeah, they're quantitative. They're highly quantitative and dimensional. We see great variation along these different measures. Are they associated with the illness and with the outcome? In fact, they seem to be associated with core traits, but importantly, it seems to be the biology that is most strongly associated with those core traits. Are they independent of clinical state? Well, I haven't really showed you any data on that. What we do know is that some of the measures that I'm discussing, part of the reason that we focus on them is because there is literature. There are literatures on these different measures to show that they are highly stable over time. They are highly reliable both across AM, PM, and different states, including within schizophrenia states of psychosis. Are they heritable? I didn't show you data on heritability. Really, the only way that we can really measure heritability is through twin studies. And in fact, twin studies have shown that similar motor measures as the force measures that I just showed you, as well as the ocular measures, there are high levels of heritability of basic sensory motor and cognitive control abilities. So it suggests that they would be good endophenotypes, but to be clear, I haven't shown any data on that as of yet. And then are they evident in unaffected relatives? Well, we have some preliminary evidence that these same dimensions are affected in parents and siblings of autistic individuals, but we wanted to replicate those findings in a much larger cohort and then look at the extent to which they're interrelated amongst family members. All right, so we're at the fourth quarter, but I'm going to fly through the fourth quarter, which is a shame because this is the newest data and I think some of the most exciting, but maybe we can have a little bit of discussion around it as well. So this is a family trio study that was supported by the Department of Defense that we conducted several years ago. And the idea was that we were going to study both in autistic individuals as well as their biological mom and their biological dad. Okay, so then we can look at interrelationships across different parents, see if there's any differences in paternal versus maternal inheritance, things like that, or interrelationships, I should say. Dads are hard to recruit. I'll just say that as a dad myself. So we got close, and there are a lot of family trios in this cohort, but the reason the math doesn't necessarily work out is because there are some dyads as well. We then identified recruited neurotypical controls, separate controls for the kids with autism, and the parents. So we have parent controls and then proband controls within the sample. I'm going to fly through some of the characteristics in the interest of showing the exciting stuff. One thing that we did differently within the study, and this is going to wind up being important, we subdivided our parents, right? So I talked before about the idea that one of the better predictors or one of the candidate endophenotypes that does show relatively higher odds ratios is background familial autistic traits or broader autism phenotypic traits within parents. And so what we did is we used a broad autism phenotypic questionnaire, which actually has published cutoffs to identify parents as being BAP positive. That is, they showed broad autism phenotypic characteristics above threshold, or BAP negative. They did not show any of these characteristics. So we did this on the individual parent level. I'm not going to show you the proband analyses for the motor stuff yet, but then we also subdivided the probands according to whether or not they had a parent with BAP positive features. All right. So when we did the force paradigms, the experiments that I just showed you, the gripping paradigm, what we found is similar to our initial study. Individuals with autism and their parents each showed increased variability over time relative to neurotypical controls. But as you look at these parents, what's really interesting here is that effect of increased variability actually is specific to those parents who did not show broader autism phenotypic characteristics. So there definitely was a moment where we said, oh darn, except we didn't say darn. But in reality, it's the BAP negative parents that seem to be affected. Why would this be the case? But if you think about that rainbow model I showed you before from John Constantino, what that's really showing is that if you start off with a case with autism and you start crossing off as not present those other candidate endophenotypes, one of them has to be there. One of them should be there, I should say. Right? And so we've crossed off for these BAP negative parents. It's not the familial autism traits that are going to be the endophenotype. And so now we're down in that model, that very simplified model, we're down to 3 candidates perhaps. And so those families are more likely to show the motor problems. And in fact, that's what we found here. It was true both for our force test, as well as we then replicated our previous family findings that saccade error. Oh, my bars are all out of whack over here. I apologize for that. I don't even know what they mean, but anyway, I'll tell you what the what the gist of this is, or what the result is. The result is that saccade error in this one. I say accuracy at top, but it's error on these figures. Error is increase in parents, but specifically in BAP minus or BAP negative parents. That is, those parents who did not show broad autism phenotypic characteristics. So it's the same story as I just told you for the force paradigm. When you look at these motor systems, the parents that are showing some motor liability or subclinical issue with motor control that is similar to what we've seen in the probands, it's the parents that don't show the broad autism phenotypic characteristics. We then we looked at the velocity of the movements, and this seemed to be actually one of our more powerful indices here, where we saw these strong effects where the differences were specific to the BAP negative parents. They showed higher velocities relative to parents that were BAP positive, as well as to controls. And then importantly, saccade velocity was highly familial. So higher velocity amongst parents, higher velocity amongst kids, okay? So we have this familial marker that is affected in autism and is affected in a subset of parents. So again, going back to this chart, what we've essentially done by saying they're BAP negative is we've crossed out this yellow box. And in this simplified model, which again is not exhaustive, but for illustration purposes, it's essentially saying that we already know that the person has autism. It must be coming from one of these 3 boxes. We're down to 3 candidates, okay? And that's what we're seeing with the motor coordination. Now, in contrast to this, we did look at cognitive control systems building upon the anti-saccade results that I showed earlier. I really got to fly here. I apologize because I'm looking at the clock. But in short, this task on the left is a stop signal task where individuals are presented with cues, a green circle, and they're supposed to make an eye movement or a button press when they see that. Sometimes, though, after the green circle shows up, they get a red stop sign. And that means even though they may have started that behavior, they need to stop it right away. So we can look at how good they are at stopping when these stop cues show up. They don't know when the stop cues are going to show up. We also looked at cognitive flexibility. This is like a Wisconsin card sort test. Individuals are selecting, they're instructed to select the correct item, and they select one of them. And then unbeknownst to them, we switch the correct item, and we look at how quickly they are able to then switch over to selecting a new item, all right? When we did the stop signal study, what we saw is that individuals with autism showed lower stopping accuracy. That is, they made more errors when the stop signal showed up. We saw that neurotypical controls really slowed down their behavior to see if the stop sign was going to show up, and individuals with autism did not slow down as much. During the reversal learning test, or the Wisconsin card sort-like test, individuals with autism were just as good as neurotypical controls in identifying the correct item or location. But when they had to switch and they started getting feedback that they were incorrect when they started pressing this, they kept pressing the same one more frequently than their neurotypical controls. They didn't switch over as well. They weren't flexible. They couldn't adapt to these new stimulus characteristics or response characteristics. So we repeated these tests within the family, a separate sample again, within the family study, and the same family sample that I just showed you. And what we found here was in contrast to the motor results. And that is, we again saw in an independent cohort that the rate of errors during that probabilistic reversal learning test, the rate at which individuals could switch to the new item, was impaired in our autism sample, but in particular in those individuals with autism who had a parent, at least one parent, with broader autism phenotypic characteristics. And in fact, the parents who showed broad autism phenotypic characteristics did worse on this particular test. They made more of those reversal errors. And we saw a similar pattern, though not quite as robust, in the stop signal test. Okay, so it was the BAP positive probands and the BAP positive parents that tended to do worse on this test of cognitive inhibition. So what does that mean? This is the last slide I promised before my thank yous. All right. So quantitative motor and cognitive control abilities. Hopefully I've convinced you of this, or at least pulled you along somewhat to the idea that they represent promising endophenotypes for understanding etiology. For understanding familial autism, inherited autism, likely representing polygenic risk. We know they're impacted in autism, they're predictive of core autism traits. They're associated with differences in nervous system function, and importantly, they appear to be affected in unaffected family members. They appear to affect different families, and that's what that family trio data shows, or that latter study, where we subdivide into BAP positive and BAP minus parents. And these two different classes of endophenotypes may be impacting or explaining cases in distinct family clusters. They may represent distinct polygenic pathways to autism. So when we talk about resolving heterogeneity in autism, at least etiological heterogeneity, it may be that we need to start understanding or identifying those endophenotypes that characterize different families. Key next steps, I'm just going to leave it there because I'm sure you have some ideas on it. But after I thank the wonderful lab that has put all this work together, in particular, Catherine has done a lot of the imaging and the motor work. Catherine Unruh, I think all of you know her, is pictured there. Robin has been working on a lot of this motor data as well. Aaron Bojanic and Shannon Kelly both have graduated and have done a lot of the family analyses for these studies. And so I did want to thank them publicly. I'm then going to go back to this slide as we have our Q&A and discussion. But thanks for bearing with me, as I know I went a little bit long, but I'm happy to take questions now. Go ahead, Nancy.
Hi, really interesting. Hopefully you can hear me. There's a little hum in my office, but
I can hear you.
okay, great. I'm not familiar with the BAP positive and the BAP negative determinations, et cetera. But is it important to do it dichotomously like that, or would you learn more if you were looking at continuously like you would with a lot of things in autism?
Yeah, I think we talked about it. I think, to be frank, the measure that we use, the BAP questionnaire, I don't think is a great continuous measure for this purpose. And in fact, that's what that came from UNC, and what that group has shown is that these categorical cutoffs are quite informative. As we plan for these studies moving forward, we're sort of developing this work around these key next steps. The current hope and plan would be to focus more on Constantino's SRS measure so that we can get a continuous distribution of those autistic traits amongst affected and non-affected individuals. So that would be the approach that I also would advocate for moving forward. Other questions?
Matt, this is Jessica Hellings, and
hey, Jessica.
Thanks so much for your great talk.
Thank you.
I might have missed because I was a bit late, I'm afraid, but do you study this only in one specific gender? Do you keep your group specific to girls or boys, since autism is often quite different in girls as opposed to boys?
Yeah, it's a good question. The samples that I showed you tended to be predominantly male samples because of case rates and what we know about sex ratios. But I'm with you in terms of both phenotype varying across sexes, and of course there are some challenges and some inconsistencies in how maybe autism is identified in females. So our current work that we've been doing over the past 5-6 years, we've tried to get closer to a 50-50 ratio, because the reality is that both thresholds, phenotypes, as well as underlying etiologies are likely to vary across sexes, but we haven't been able to dig into that within this family work yet.
Okay, thanks.
Sure thing. Yeah, Christine.
Can you hear me?
I sure can.
Okay, so going back to the ADOS, was it just to confirm their autism diagnosis, or did you look at the different levels?
Mm. When you say different levels, do you mean like within the CSS and the mild moderate? Yeah. No,
Yeah.
we didn't look at that categorically. So it's kind of the antithesis of Nancy's question, right? Because we did look at that dimensionally and just look at the CSS score as opposed to those qualitative ratings. I don't know if I would with that measure. I just think in terms of the reliability of those cutoffs, not to mention the reality is then you start really subdividing the sample down, at least the numbers that we have, to numbers that are probably too small to be highly replicable. But it could be another way to look at those in tiers in terms of, mild, moderate, severe. I will say though, that when we looked at the BAP negative versus BAP positive probands, and to be clear, the BAP positive probands are defined by either mom or dad or both, exceeded that quality, that threshold. There didn't seem to be strong differences in the clinical profiles, at least just looking at autism severity or looking at IQ. Now, this is a relatively restricted sample. We study people probably down to about an IQ of 70. So it's possible that there are distinctions. The model that I showed or shared with you suggests that in fact that it really operates under this model of allelic.
You should come inside. It's too hot for you, my boy.
Hey Jessica, if you could actually, I think I can, there you go, got it. But it would suggest this pattern of equifinality. In other words, that no matter which of these you have, it can lead to autism, but it doesn't lead to different type of autism or a different pattern of autism. But to be frank, that's a very open question. It's a great one too. So. Yeah, Megan.
So I guess my question is slightly related to Christine's in that I'm also thinking about if you've looked at any sort of differences, if you kind of try and separate out features more related to restricted and repetitive behaviors versus the social communication aspects.
Yeah, you mean if we look at phenotypically, like if children, if probands vary as a function. Yeah, and so based on the behavioral data that we've done with the probands, the hypothesis there would be that these BAP positive families or the families that show these cognitive control deviations, either inhibitory control or the cognitive flexibility, that those would be the families and individuals that are showing more severe repetitive behaviors, because we saw associations between those tests and clinical repetitive behaviors. But to be clear, those clinical associations, they're kind of variable across our samples. I mean, sometimes they're like R's of 0.2. When we initially looked at in that first anti-saccade study when I was a postdoc and everything was working out, right? That lasted for about a year. Like that R was like 0.6, and we haven't seen an R that strong since. So based on that background data, I would hypothesize those BAP positive families, or at least those BAP positive families that also show the cognitive control deviations, may have children who show more severe restricted and repetitive behaviors. But again, that's something that we're really going to have to, the goal here is that we're going to be building a larger R01 plus level study, because we're going to have to look at this in hundreds of families.
Yeah.
Great question. And then, yeah, Mohammed, I think I saw your hand up. Go ahead. Oh, you're muted though.
I was trying to find a way to raise the hand over.
Sorry,
but I missed that. I have a question about I'm not well aware of BAP positive, BAP negative, how you define them, but just for curiosity, how in case when you would find variants, like it could be common variants or de novo variants in these individuals who have autism, and then in the endophenotypes, when you see that those variants in those parents, so how can we differentiate the endophenotypes in the parents compared to the controls who are completely unrelated to the people who have autism?
I'm not sure I quite understood that question. I thought you were going different direction. So you're trying to differentiate the endophenotypes in the parents, but versus the controls, or?
Right. So because when when we are looking at the variants, we really want to find out the significance of those variants when using the control populations, right? So
Right.
those control populations, if they are well defined quantitatively
Yeah.
for the similar endophenotypes, that would help, right? So my first question is, let's assume if you was able to identify a de novo variants,
Mhm.
de novo variant in the people who are autistic, and then because those variants would not be
I see.
inherited from either parents, and then how would this endophenotype help to identify the significance of those endophenotypes with those de novo variants with the endophenotypes?
Yeah. So I mean, I guess we did look at it for a large number of these families. Like they came in, we got blood from a lot of these families, and we looked for and actually exclusionary were a lot of those either de novo or highly penetrant variants. So what we refer to as kind of single monogenic autism, which is kind of a misnomer, but thinking about these highly penetrant genes, and so most of those individuals don't get in. The other thing is those de novo variants as well as those highly penetrant variants are most commonly also associated with IDD. Right. And so it is rare that we would see those de novo variants in an autism case without IDD. Right. So there is that fact. That being said, it's possible that there are individuals in that study who we didn't detect, who we didn't identify, and they don't have a comorbid IDD. That with those individuals, we would essentially, you may expect to not see strong traces of these endophenotypic markers in parents. But that being said, you have to consider the fact that there's significant pleiotropy, right? So there's pleiotropy associated with those different de novo variants. And that is to say that some individuals who have those variants have IED, some have speech language impairment, some have autism, some have all of those, some are unaffected, right? And so for those individuals, and there is actually some data on this, that some of these common variants or endophenotypes may push those individuals with those rare variants into autism, right? So in fact, the rare variant is not enough for the full phenotype. And so you could expect to actually see some of those endophenotypes or those deviations within those families and within those individuals. But I hear what you're also saying in that these endophenotypes, some of the beauty of it is they're quantitative, and frankly, these traits are distributed across the population, right? And there's overlap within our families and even within our probands and the neurotypical population. And so frankly, what needs to be done, I think I have this, I don't think I said it explicitly in the key next steps, but the ideal study of this is really is a large scale study looking at these traits across an unbiased population and determining whether they predict at the parental and then at developmental levels, they predict autism traits or autism outcomes across the population. So this is actually where we're going with our current project if we could get it funded. But the idea here is that we would really engage in what we refer to as phenoscreening, is that we would start screening individuals through parental traits as opposed to ascertainment of autism cases and non-cases. And then you would get that full, you would cover the full population and be able to look at those traits across the population of affected, unaffected, and variably affected in between, which we're missing oftentimes. So I think that hopefully that answers your question.
Yeah, thank you.
All right. Thanks for coming. All right. Everybody, thanks for staying on and thanks for having this discussion. I look forward to seeing some of you tomorrow morning at ProSem, and the rest of you, I'm sure I'll see you soon, at least in 2 weeks at our next talk. Good seeing you all.